protoplast, whatever it may be. Let's see just brief introduction about the methods of gene transfer or genetic engineering. The transfer of desirable gene from one organism to other organism and expression of foreign gene in the genome is referred as genetic transformation. So what is gene transform? Transform means we select a desired gene from one organism and introduced into the another, simple. Introduced into another organism. So this is called genetic transformation. Genetic transformation is a powerful tool and an important technique for production of transgenic plants. This is useful in introduction of foreign genes into crops. The foreign gene actually other than other species, other desirable character species. When you introduce into a crop, it creates new genetically modified organisms. The plant breeders and genetic engineers share the common goal of plant improvement. While the plant breeder traditionally use selective breeding for varietal enhancement, genetic engineers continue to develop technique for the isolation and insertion of the gene for desirable traits. If you take the traditional method actually takes a lot of time to get desirable character. But through this gene transfer method, desirable gene can get within short period of time. In these techniques, genes was unavailable to particular plant species, may be obtained from other organisms, such as plants, microbes, or any organism, using both molecular and cellular biology techniques. So this is the brief introduction. And about and one more thing, the plant transformation was first discovered in tobacco plant in 1984. Since then, rapid development in transformation technology have resulted in genetic modification of many plant species. We have seen the number of species like golden rice, potato, variety of potato, brinjal, BT brinjal. They are all we get by gene transformation method. Various method for introducing diverse genes into plants. All the transformation techniques involves uptake of DNA and delivered into the plants. Gene transfer methods in plants make use of variety of target cell types. These such as maybe include protoplasts, cultured cells, meristem cells, immature embryos or embryos pollens. These are the targets to introduce the foreign DNA. Let's discuss about the types of gene transfer method. Gene transfer clear. If the gene transfer means transfer of the desired gene from one organism to another organism. Then how to transfer? What is the requirement for the transferring of the desired gene to one to another? So there are two methods to transfer the desired gene. One is direct transformation. Second one is vector mediated gene transformation method. First one, direct transformation. In direct transformation, desired gene directly introduced without any vector. These genes doesn't carry any vector. We can directly introduced into the another plant cells. Second what type is vector mediated gene. In this type, vector should involve. The vector may be bacteria, bacteriophages, virus, whatever it may be. The vector plasmids, they are all vectors. First, let's discuss about the direct method of gene transfer. Direct method of gene transfer. Just wait. Right. In direct method of gene transfer, the first of all, what is direct method? The term direct or vector-like transfer of DNA used when the foreign DNA is directly introduced into the plant genome. Direct DNA transfer methods just on the delivery of naked DNA into the plant cells. So in this direct method, several different methods are present, such as electroporation method, microprojectile method, second one, and micro injection method, liposome mediated method, and some chemical gene transfer methods. And let's discuss first one, electroporation method. So in electroporation method, we selected the cells which want to introduce the desired gene. Just let's take the pointer. So these are the target cells. These are DNA fragments, desired DNA. So you selected the target cells and the desired DNA, desired genome. Both. This both mixed together in a vessel, cells with DNA. This is the A is the cell. The cell from the A cells separated the DNA fragments. After that, to this vessel, give the electrical pulses. Electrical pulses. So after getting electric pulses, due to the electrical force pulse, the cells become weakened and they make it whole to plasma membranes and cell wall and nuclear membrane, make it whole. Later, the desired DNA enter into the cell and enter into the nucleus, finally incorporate with the DNA, the cell genome or cell DNA. This is the process. So first of all, what is electroporation? So introduction of foreign DNA, foreign DNA into living cells, these cells by exposure of brief electrical pulse. Exposure of brief electrical pulse is known as electroporation. In this techniques, mixers containing of cells and DNA, foreign DNA is exposed to very high voltage electrical pulses. This high voltage electrical pulse may be 4,000 to 8,000 volts. 4,000 to 8,000 volts. 4,000 to 8,000 volts. 4,000 to 8,000 volts. It results in formation of transient pores. As a result of due to this electrical shock, electrical force, the make it the holes, some pores in the plasma membrane. Through these pores, the which the desired DNA enters inside the cell, and the nucleus later finally it is entered the nucleus, finally fused with the nucleus genome. This technique can be used for delivery of DNA into intact plant cells and protoplast. Intact plant cells means the healthy cells, healthy protoplast only. Right? So this is the first method. And second method, microprojectile or particle bombardment gun or biolistic method. Let's see the second method. Right. Particle method or another name microprojectile bombardment gun or ballistic method. So just 1 minute. Sorry for the inconvenience. Right. So in this method, it is act as a gun. So first of all, in this method, foreign DNA containing desired gene that is coated with into surface of tungsten or gold particles. It takes the right. So firing pin. This is the firing pin. This is the foreign DNA coated with foreign DNA. The blue color, sorry, coated with coating of the foreign DNA. The coated with here the chemicals using this coating, tungsten or gold, tungsten coating, maybe tungsten or gold particles. This is called microprojectile and are carried by macro carriers, macroprojectile. Nylon, tungsten, nylon, whatever, microprojectile. So this is the instrument. This macro carrier, this is macro carrier, carries inserted into apparatus and bombardated very to high velocity onto plant cell tissue. With this, the DNA segments are released which enter the plant cells and integrated the genome. So this is the macro instrument. This is the macro instrument. Here the micro projector, this one. This is firing pin. Black charge is like a piston. If you press this, the pressure falls on the black charge. The micro projectile pushed the micro projectile. It is forcefully already we settled just base of this instrument. One plate containing the target tissue. This is the target tissue. The target tissue this target tissue placed the just below the instrument. When you apply the force, the high velocity create high velocity to the plant tissue. Due to the force, it is forcibly enter into the tissues. So this is bombardment method. Bombardment method mainly desired gene coated with tungsten or gold, right? But the target cells, the target tissue placed the below of the instrument. After applied the gun firing, like it is high velocity create and it forces the target tissue into the target gene into the tissues. So this is particle or bombardment gun methods. And next one, micro injection method. Micro injection. Right. This is micro injection. This is depend on high technical value, high skillful. Only skillful person can do this. So in micro injection method, actually this is the target cell. So microinjection involve direct mechanical introduction of DNA into the nucleus or cytoplasm. That means the foreign DNA directly introduced into nucleus or cytoplasm. During the glass micropillary, this is glass micropillary, glass micropillary using glass micropillary injection pipette having fine tip. In this technique, target cell is positioned under the microscope because we should observe where the cytoplasm, where the nucleus. We keen observation, we doing creep observation. That's why this is carried out under the microscope with a direct inject into the cytoplasm or the nucleus with help of the micro manipulator. This is called micro manipulator. The one micro manipulator holds the pipette. One micro manipulator holds the pipette and another holds the micropillary needle. This is needle. One is hold pipette micropillary injection. Micropillary injection. One micro manipulator holds the one micro manipulator holds the pipette. Which pipette? Glass micropillary injection pipette. This one. This hold for one micro manipulator. Second one, this one target one, hold with, and another holds with micropillary needle. This is needle. Micropillary needle. While the gene transfer is done, the recipient cells are kept immobilized in agarose embedding and held by suction holding pipette. Once the gene is delivered, the injected cells then cultured into in vitro and regenerate into transgenic plants. So in this method, first one, actually two instruments mainly utilized here, the glass micropillary injection. Second one is this is the target cell mostly without cell wall. We are taking the cytoplasm or nucleus. So this is nucleus. Now, actually this all process under the microscope. Here the one hand hold with micro manipulator. This is hold with micro manipulator and needle hold with another micro manipulator. After that, where the desired genes are present inside this micropillary injection. This micropillary injection. When you press this button, this desired gene enter into the either the cytoplasm, or if you want to inject the nucleus directly, this micro injection suck into the nucleus. This micro injection suck into the nucleus and release in the nucleus. Later, this desired gene and these both genes are incorporated. Even if you release it cytoplasm also, after that, these both genes are fused. So this is the micro injection method. Micro injection method, is it clear? So micro injection has achieved only limited success in plant transformation due to thick cell wall of plant and lack of availability of single cell to plant regeneration system in more plant species. And also the gene gun method also the not this not appropriate, because when we use the high velocity, due to the high velocity force, there is a chance to destroy the tissues and destroy the genomes also. That's why the micro injection, mainly the micro injection, it is based on skillful process. That's why these these both are shows the limited success to generate the transforming cells. And next one, liposome mediated transformation. See the next one. Yeah, liposome mediated transformation. The liposomes are actually, these are the chemicals. The liposomes are artificially created microscopic lipid vessels. The containing phospholipids membrane, contain phospholipids membrane. Liposome containing plasmid, liposome containing membrane, plasmids. Phospholipid membrane. These are produced when phospholipids are dispersed in aqueous phase. In this method, the DNA enters the protoplast via endocytosis of liposomes. This is the protoplast target. When the both are fused, this is liposome containing plasmid, protoplast. When both the fused, due to endoosmosis, that means the process of osmosis, the plasmids in the protoplast, release plasmids in the protoplast. This is based on endoosmosis process. Endocytosis of liposomes. Endocytosis of liposomes. Generally, this process involves three steps. The first one, addition of liposome to the protoplast surface. This is the first addition. Addition means nothing but attachment of liposome protoplast surface. After addition, fusion of liposome at the site of addition and release the plasmid inside the cell. This is cell, black spots plasmid. The release of plasmid inside the cell. Plasmid. After attachment, this is first one, addition, attached, release of the next, finally release of the plasmid. Liposomes being positively charged tend to attract negative charged DNA and cell membrane. In this process, the engulfed DNA is free to be integrated in the host genome. However, there have been a very few successful reports on the application of these techniques in plant science, like gene gun method, like microinjection method. This is also have low success rate. So this is like liposome mediated transformation. Next one, chemical method. So chemical gene transfer method mainly used by polyethylene glycol mediated transformation. The second one, calcium phosphate mediated transformation. First, polyethylene glycol PEG mediated transformation is a method used to deliver DNA using protoplast as an explant. This PEG method, the polyethylene glycol method, only utilized by protoplast. Only protoplast, inside the protoplast. The polyethylene glycol in the presence of divalent cations, C calcium, destabilize the plasma membrane of protoplast and it permeable to NAC DNA. In this way, the DNA enter into nucleus of the protoplast and finally get integrated with the genome. The method is similar to the electroporation method. It's look like electroporation. What happened in electroporation? Electroporation also some pores are formed. Here also some pores are formed, but in electroporation by electric force. But here by some chemicals. For example, polyethylene glycol, polyethylene glycol, calcium. When we apply this, for example, this is plasma membrane. When we apply this plasma membrane distable like a small portion formed like that. After that, when we mix it with the desired genes, Now desert genes enter into the center. The cells are nucleus. Finally, the genomes are integrated. The method is similar, okay. Of course, these are similar to electroporation DNA to be introduced is simply mixed with protoplast. And uptake of DNA situated the addition of polyethylene glycol rather than electrical pulse. It not involved any electrical pulse. Instead of electrical pulse, only polyethylene glycol used. The polyethylene glycol mediated transformation has several advantages because it is used to handle and no specialized equipment is required. No pulse, no specialized equipments, they are all not required. Just only the one chemical enough. That's why it is easy to introduce the desired GNA. And also it shows the most success rate. PG mediate transformation has several advantages. However, the technique is rarely used due to low frequency of transformation. But the frequency rate low. Of course, it is success rate okay, but frequency is low, frequency of transformation low. That's why it cannot be regenerated into the whole plants from the protoplast. Even though using this method, transgenic maize and barley plants, the polyethylene glycol method mediate transformation, a large number of protoplasts can be simultaneously transformed. This is one advantage. This technique can be successfully used for wide range of plant species. So, polyethylene glycol. Next one, calcium phosphate partition. The DNA is allowed to mix with calcium chloride solution and isotonic phosphate buffer to from DNA calcium phosphate precipitate. When the actively dividing cells in culture are exposed to this precipitate for several hours, the cells get transformed. The success of this method is dependent on high concentration of DNA and the protection of complex precipitate. So this is the calcium phosphate precipitation mediated. So these are all direct method. These are all direct method only. All right. So in direct method, no vector used. So in this method, first let's summarize. Electroporation method. In electroporation method, the plasma membrane gets poured with help of some electrical pulse shock. Next one, bomber and gene gun method or biolistic method. In biolistic method, desired gene coated with tungsten and gold particle inserted into another instrument like gun. When we apply the force to the velocity force, the desired gene forcefully introduced into the tissues or cell. Next one, micro injection method. In micro injection method, with help of the micropillary injection pipette, and actually this is carried out under the microscope. We need micropillary injection one, another one is needle, and another micro manipulator. Micro manipulator, the instrument used for the holding of the micropillar and needle. Later inject into the whether it is nucleus or cytoplasm, whatever it may be. After injected into the cell, injected into the nucleus or cytoplasm, finally the desired gene get incorporated with that. Liposome mediated. In liposome mediated, liposome contain plasmids. The three steps first, they are their addition. They are first of all in three steps. First, addition of liposome to the protoplast. After addition, fusion of liposome to the that closely fusion to the where the addition takes place. The finally release of plasmid into the cell. Next, a chemicals method. In chemical, one is polyethylene glycol method, one is the calcium phosphate precipitate method. So these are the direct gene transformation methods. Let's see the vector mediated gene transformation method. Right. In vector, so it name itself vector. Here the vector most probably using agrobacterium. Using agrobacterium is the vector. Agrobacterium tumefaciensis, the vector. This is known as natural genetic engineering. Naturally gene transfer. That's why it is called genetical engineering of plants, because bacteria have natural ability to transfer the DNA or desired gene of their plasmid into the plant genome. This bacteria enter into the plant cell at the wound site and cause an unorganized growth of the cell mass of cells group. This group of cells called as crown gall. So Ti plasmid are used as gene vector for delivering target plant cells and tissues. The foreign gene is cloned in the tDNA region of Ti plasmid in place of unwanted sequence. Let's know about the natural genetic engineering agrobacterium. The agrobacterium tumefaciensis is a soil-borne gram-positive, gram-negative bacterium, and it is the rod-shaped. We can see here the rod-shaped and mobile. Mobile means with flagella also present here like this. Flagella also present. Mobile and belongs to the bacterial family Rhizoidae. So Rhizobiaceae. So this is the brief introduction about the bacteria. So there are two types of plant pathogenic bacteria which are used in natural gene transfer in plants. One is Agrobacterium tumefaciens that induce the crown gall disease in plants. Second one is Agrobacterium rhizogenis that induce the hair-eated disease in plants. First discuss one crown gall disease of Agrobacterium. This is the now crown gall. Crown gall. Right. Before that, we should know about Ti plasmid of bacterium. It's Ti plasmid of bacterium. Ti plasmid of bacterium. Right. So Ti plasmid of bacterium. It is a independent replicating circular DNA. You can see here the circular like this circular DNA. Circular DNA. The Ti plasmid of agrobacterium circular. Okay. It may be 200 kilobytes. 200 kilobytes in length. The tDNA, transferred DNA is variable in length from range from 12 to 24 kb, which depend on bacteria strain. Right? Ti plasmid as major region for virulence, origin of replication, oncogenicity. You can see here, this is virulent region, with gene. Virulent region. Second, origin of replication. Here, green color. Go for origin of replication. Oncogenicity and catabolism, and next one, oncogenicity. and open catabolism. Let's take oncogenicity, open catabolism, oncogenicity and open catabolism. And apart from that, some TDNA region, left border, this left border, 25 base, auxin region, cytokinin region, open region, common. Okay, open region. They are all present. First, TDNA region. This is all TDNA region. So TDNA region has the genes for the biosynthesis of auxin, and cytokinin. Biosynthesis for auxin and cytokinin and opines. These three. And is flanked by the left and right border. Left border, this is right border. These three genes, auxin, cyto, and aux, are referred to as oncogenes. These are all together called as oncogenes. Opine, cytoxin, auxin, these three called as oncogenes as they are determined of the tumor phenotype. You know what is the function of auxin and cytokinins? Auxin and cytokinins involved in morphogenesis. So, when the presence of auxin cytokine in extra ophine present, it leads malformation. It will appear phenotypically tumor. TDNA border is at 24 kb, 24 kilobase sequence present at the either side, right and left. So here let's discuss one by one portion. First one, just wait. Different genes present in TDNA region. Where the I am. Okay, no need, no need. This is enough, right? Right. So mainly here the only one we need the T region, TDNA region, because the TDNA region produces the auxins and cytokinins and opines. With presence of all this, morphogenesis takes place, malformation takes place as a result formation of crown. That's gall formation, like a tumor. So that's why it's called as a gall ground. First, virulence region. This region responds for the transfer of tDNA into the host. tDNA, this tDNA transfer into the host. This is host cell. Transfer into the, just let's change the color. Host cell. So here the gene responsible for the transfer of tDNA. This is tDNA or T plasmid, which is present in bacteria, agrobacterium bacteria. So how the agrobacterium T plasmid enter into the host cell because of virulence region. This region, virulence region only, and is referred to as vir or virulence region. The vir region codes for protein involved in tDNA transfer. Okay. In this region, some of the different type of operands have been identified. These are vir A, vir G, B1, C1, DI, D2, D4, and vir E and E2. So this is the first region. Second, open catabolism region. Open catabolism region. This region codes for proteins involved in the uptake and metabolism of opines. Besides the above three, there is a region that responsible for the origin of DNA replication. That means uptake of metabolic opines. This opines. Now this is okay, right. After get activated, the ORI site started. Origin site started. Next, DNA transfer and integration onto the host cell. In this step, how tDNA transfer and how to integrate it. First step, in the process of tDNA transfer and integration of the host cell, given below step by step, the first signal induction of agrobacterium. First step, this is the first signal induction agrobacterium. Second step, this agrobacterium attached to plant cell. Second step, attached to plant cell. Production of virulence protein. Production of virulence protein. These are production of virulence protein after attachment. Production of tDNA strand. Next, transfer of tDNA of agrobacterium with help of virgins. Transfer of tDNA into the plant cell integration. That is the final. So first, let's discuss one by one. First, signal induction of agrobacterium. For example, this is the wounded plant cell. This is the plant cell, wounded plant cell. First, the wounded plant cell release some phenolic chemicals to out. Phenolic chemicals. Phenolic chemicals and sugars, which are recognized as signals. These signals induce the result in the sequence of biochemical events in agrobacterium that ultimately helps in transfer of tDNA of the plasmid. When the plants get wound or injury, immediately the plant release some the phenolics and sugars. So this phenolic and sugar, the soil-borne bacteria, mainly agrobacterium, get attracted towards the plant cell. Next, attachment of agrobacterium. When they are attracted and reach toward the wound or injury part. Second, this agrobacterium attached to the plant cell through the polysaccharide, particularly cellulose fiber produced by the bacterium. Several chromosomal virulence genes responsible for the attachment of bacteria cell to plant cell. Several chromosomal virulence genes which are involved for attachment. Next, production of virulence protein. As the signal induction occurs in the bacterium cell attached to plant cell, a series of events takes place that result production of virulence protein. By the signal induction of phenolics stimulates viriae. Next, virilis from the virulence region. Viriae, virci, both are released. This induces the expression of virulence genes of Ti plasmid to produce the corresponding virulence protein. So finally, viriae, virci activity, the virulence proteins are released. Next, production of tDNA strand. The right and left border of tDNA, this region. tDNA, this region. Right and left border of tDNA recognized by vir D1, vir D2, which are released from the virulence gene. These proteins are involved in the production of single stranded tDNA. Single stranded tDNA, which gets attached. The transfer of tDNA agrobacterium. Right. After formation single stranded DNA, the single strand DNA and virulence D complex in association with the Vir-G exported from the bacterial cell. Already told you the virulence region importance. Virulence region importance introduced of the T plasmid into the host cell. So, but here after formation virulence D or G, this both with help of this both the single stranded tDNA exported from the bacteria cell into the host cell. Like transfer of tDNA into the plant cell and integration. After enter into the host cell, in the plant cell, the tDNA gets covered with virulence E2. This covering protects the tDNA from the degradation of nucleus because already this is the nucleus in plant cell nucleus. If the tDNA present, single stranded tDNA, this is single stranded tDNA present. When entered the nucleus, already inside the nucleus, nuclease enzyme there. What is the function of nuclease enzyme? If nuclease enzymes breaks the this rDNA, sorry, breaks the or TDA, breaks the tDNA, right? But to prevent this, Vir D2 and Vir E2 interact with the sorry, where it is. So to prevent this, tDNA gets covered with virulence E2. This is act as a protection layer. The tDNA from degradation of nucleus. D2 and Vir E2 interact with the variety of plant protein which influences tDNA transport and finally transport and integration. The plant protein complex enter the nucleus through the nuclear pore complex and get integrated to plant chromosome. So, finally, this single stranded tDNA attached to the plant cell nucleus genome. Now it becomes recombinant. So this is about the crown gall disease of agrobacterium. Right. Next, airy root disease of agrobacterium. Airy root disease of agrobacterium rhizogenesis. Same look like a crown gall disease. The process. But here the different is vector. Just 1 minute. Sorry for inconvenience. We will discuss later. Tomorrow, I will complete the hairy disease of agrobacterium, and also uh, actually tomorrow last class. The only I'm going to take the one important class, but that is the 11th unit, improved growth. Okay. Thank you all.
The number you have dialed is